Welcome all, and thank you for joining today's webinar. My name is Robin Dixon and I'm a senior viticulturist with the AWRI. In the spirit of reconciliation, the AWRI acknowledges the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respects to their elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. I would also like to acknowledge Wine Australia for providing funding and support for the webinar program via the AWRI Extension Project. In this session, we'll look at breeding and evaluation of new mildew-resistant wine grape varieties for Australian conditions. But before we jump in, I'd like to make a few reminders for people who are new to AWRI webinars. So if you'd like to leave a comment or question during the webinar, use the Zoom toolbar down below. You can see the Q&A section there. You can just add your questions, press send. We'll be holding the Q&A session at the end. But feel free to put any questions in there at any time throughout the webinar. A reminder also that the webinars are recorded and a link to the recording will be emailed out to you after the webinar. So for anyone who has just jumped on, welcome. Today's webinar topic is breeding and evaluation of new mildew-resistant wine grape varieties for Australian conditions. And it's a great pleasure to welcome Dr. Ian Dry from the CSIRO and Ben Harris from Treasury Wine Estates. So first up, we'll have Ian. So Ian Dry has been actively involved in plant research for more than 35 years in Australia and the UK in areas covering plant biochemistry, molecular plant pathology and plant breeding. He currently leads the CSIRO research team focused on the development of new wine grape varieties with resistance to fungal pathogens. So Ian, if you're ready to go, if you can please share your screen.
Share my screen.
Thank you. Wonderful.
How's that look?
Yeah, that looks great. Thank you, Ian.
All right. Thanks, Robin. Thanks everyone for joining today. I've just put another title slide up because I wanted to acknowledge the collaboration. This is a joint project between CSIRO and colleagues in New South Wales at the Department of New South Wales BPI and at Charles Sturt University. So I just want to make sure I acknowledge them before we start. So today what I want to give you an insight into is give you an overview of the sign breeding program. We also have a rootstock breeding program here at CSIRO, but I'll only be talking about the sign breeding program today. And to give you an insight into the challenges and considerations that you have to take into account when developing a strategy for a breeding program like the one we're doing. And the first thing to say is that our focus for the sign breeding is on the two major diseases, powdery mildew and downy mildew, for a number of reasons. One is they are the most economically important or impactful diseases of Australian viticulture. And here are the numbers: 92 million a year for powdery and 76 million a year for downy, which was based on an estimate done by a GWRDC project back in 2010. So that indicates the cost to the industry, both in terms of spray application and also loss of yield and quality. But that doesn't actually tell the full story, because it doesn't take into account issues to do with environmental impacts of the spray. So think about all the carbon emissions from operating the spray equipment, and also the negative impact of these sprays on the beneficial insects in the vineyard. And of course, it doesn't take into account the ongoing problem we have with the development of isolates with resistance to a number of the common organic fungicides that are being used. And SARDI and AWRI have a project looking at that at the moment. So it really is a major problem. And the other thing I would say is that the other reason for focusing on downy mildew and powdery mildew, as distinct from something like Botrytis bunch rot, is that through evolution, plants have actually been very successful in evolving genetic resistance against the mildew type pathogen, but have been spectacularly unsuccessful in developing resistance against necrotrophic pathogens like detritus. So that's why these are good targets for breeding as well. Now, the fact that plants had evolved resistance to mildew pathogens was known, way back in the 1880s, and breeders actually started back then trying to introduce the resistance that had evolved in the wild North American grape species into the existing European or French varieties. And they were successful in doing that. They were able to produce what are termed generally the French-American hybrids. These hybrids did show resistance to mildew, but unfortunately, they had very poor wine quality because many of these wild species have genes that impart negative traits on the wine. And these French-American hybrids really gave hybrids a bad name. And now you talk to people in the industry about your breeding hybrids, they're very skeptical about whether you can actually get good quality with those hybrids. Now, the problem with the hybrids back in when they initially did these crosses is they were only doing one cross, and they were producing either what we call an F1 hybrid or perhaps doing one other cross. And when they do that, half of the genome is coming from the French variety and half is coming from the mildew-resistant variety. So you are obtaining the resistance from this wild species, but you're also obtaining all the negative quality genes. The breeding program that we're involved in is fundamentally different in that we're using different types of hybrids, and we're using hybrids that have undergone what we call a back crossing program. And this back crossing program is very common in other crops, but it's not so common for perennials, particularly grapes. And it wasn't until Alain Bouquet, a breeder in France in the early 70s, actually took an F1 hybrid that had been created in North Carolina, actually in 1916. He imported that into France and he started a back crossing program. So the idea of the back crossing program is you carry out a series of crosses with your elite variety, and every time you do that cross, you reduce the contribution of the wild species by approximately half. But in every generation, you select for those progeny that still contain resistance. And so as you do that, you gradually reduce the level of the wild species, and you end up with something which is essentially, here we are at back cross 5, it's 98.4% vinifera and only 1.6% Muscadinea rotundifolia. But in that 1.6%, you still have the powdery and downy mildew resistance. And what Alain was able to show was that Muscadinea rotundifolia has what we call a single dominant locus that confers resistance to both powdery mildew and downy mildew. And that locus was called the Run1 RPV1 locus, based on resistance to the Latin names of the two pathogens. Now, we actually subsequently cloned this locus and sequenced the genes here, and we're able to show that there were two genes at this locus. One indeed conferred resistance to powdery mildew, and one conferred resistance to downy mildew. And we actually made transgenics, but because of the moratorium on the use of transgenic grapevines, we couldn't go any further with that work. So we had to go back to looking at developing new varieties using more classical breeding techniques. But we have introduced some more modern techniques in doing that. So essentially, we took the backcross 5 progeny that Alain Bouquet had developed. We imported that into Australia, and then we carried out crosses with 8 premium white and 8 premium red varieties and generated about 8,000 seedlings. Now, obviously, that's going to be an enormous amount of effort if you have to plant every one of those in the field and evaluate them. What we can actually do is use something called marker-assisted selection by taking a leaf of the germinated seedling, extracting the DNA, and then screening that progeny for the presence of the genes that we want to have been inherited. And we can cut that down, in this particular case, we cut that down to 1,200 seedlings. And so these markers are DNA probes, if you like, that indicate whether you have inherited the particular genes of interest. So of course, we wanted to identify those progeny that had inherited both the Run1 and RPV1 resistance genes. We only wanted to keep those grapevine progeny that were producing hermaphrodite flowers, because they're more likely to produce fruit than those progeny that only have female flowers. When we're crossing with a red variety, it will segregate for white and red grapes. So we used another marker to find those grapes that only produce red grapes. And also within the white varieties, some of those are muscat producing varieties like Muscat Gordo Blanco or Frontignac. And we wanted to pick up those that still had the muscat production potential. So again, we have DNA markers that can tell us that that fruit is going to produce muscat when it's mature. So those 1,200 seedlings were planted in unsprayed blocks at Nuri and evaluated for disease resistance and vine performance. And some of you who have come to my talks before have seen this slide before. This was in the very bad year 2010-11. So these are our new resistant varieties which have been unsprayed for the whole season, no disease at all. And this is a bunch that had been taken from an adjacent row that had been sprayed during the season, but still showed a lot of disease at the end of the season. So we've never seen any disease on any powdery or downy on any of our plants in the field since they've been planted. So they do appear to be strongly resistant. So having looked at the viticultural characteristics like yield and disease resistance, bunch architecture, those sorts of things, the other major thing we had to do, of course, was to look at wine quality. The only way we can really do that without generating huge numbers is to do single vine ferments, which obviously has its limitations, but it's the best we can do. So after about 3 or 4 years, we can get enough fruit on the vine to carry out these single vine ferments, both reds and whites. And then we relied on the cooperation of commercial winemakers from a number of companies to help us try and evaluate the wine quality in inverted commas, a relative wine quality, if you like, as to which of those 1,200 varieties were producing grapes that could potentially make interesting wines on a commercial sense. So the original analysis and evaluation took place at Nuri, and we started with 1,200 vines planted in 9-10. And from those, we reduced those 1,200 down to what we call our 20 best reds and whites. And so they were selected on the wine scores of the commercial winemakers from single vine ferments, on consistently high yields, and also on good acid pH at harvest. The next stage we realized was going to be really important was to replicate those 20 best reds and whites to get more wine to be able to do small or medium scale wine analysis. But also we believed it was important to try and replicate these in different regions of Australia and grow them under different conditions as proxies for the different regions. So for example, we have a field site, CSIRO has a field site at Irimple, so that's a good proxy for the Riverland and the Sunraysia. We're collaborating with our colleagues at Wagga, particularly because they have a winery there for the small scale winemaking. And I know Wagga's not a classic wine growing region, but it's a good proxy for the Riverina. And then later, we realized we would like to evaluate the varieties in a cooler climate. So we planted a number of these reds and whites on New South Wales DPA property in Orange to see how they perform under cooler climates. And so these were grown for a number of years. And again, we get more information on yield and particularly get enough fruit to then make wine from those for small scale winemaking evaluation. I should say all of the wines have been made at the CSU winery. So that has required us to actually truck fruit from Irimple overnight in refrigerated trucks to Wagga, where they can be used for fermentation, because we thought it was really important, if we're going to do the comparison, we really need the winemaking techniques to be identical in each case. So what do the results show? So I don't have time to go through all the results today, but what I'm showing you is some of the viticultural and wine data from, in this case, the reds. So the 20 reds are listed down here. And the other thing I'll point out is that we also, in the last couple of years, we've realized it's important to actually carry out some control ferments with classic premium varieties that have been grown on the same property under the same conditions, and from which wine has been made with exactly the same procedure. So the first thing to point out is that of the 20 reds that were selected through the process, actually a large number of them have one parent, that's Dunkelfelder. And interestingly, Dunkelfelder is a red flesh variety. And not only are we getting really high levels of anthocyanin production with these progeny, but through the wine evaluation, the progeny that had Dunkelfelder as a parent seemed to perform very well in terms of quality, other quality parameters as well, flavor and aroma. Now you can see the impact of having Dunkelfelder as a parent if you look over here at the wine anthocyanin. So normally the Cab Sav and Shiraz grown in the property, it's around 100 to 200 mg per litre, but in the wines made from Dunkelfelder, you have much higher levels. So this we believe is a real selling point for these wines in terms of the really high levels of anthocyanins. Those coming from Shiraz and Tannat have levels which are more akin to the normal Shiraz and Cabernet. And the same is for those grown in Wagga as well, really high levels of anthocyanins. Now, what we wanted to do is we carried out tastings of these reds and whites with a number of different panels, some winemaker plus grower panels in different regions. And what we've been looking for is trying to get an idea of how the wines rate relative to classic premium varieties and how they score across different regions. First, the first thing I want to draw your attention to is these are the scores of blind tastings of our varieties and the control varieties here. And if you scan through here, I think you'll find that both the wines made in Rimple and Wagga, that our wines actually stack up pretty well in terms of scores. And as I point out, I stress again, these were blind tastings. And so our wines actually score as well, if not better, than the classic varieties when made this way. They're still only experimental wines, but I think that gave us a little bit of comfort that it was possible to produce hybrid grapes that would potentially produce good wines. Just moving on to have a look at, that's also the case for the whites as well. In the whites, of the 20 that were selected, we have 4 different parents, Muscat varieties, Frontignac and Muscat Gordeau Blanco, and then Verdelho and Riesling. So these are the ones that came through the tasting procedure. And again, you can see in a blind tasting of our wines compared to Riesling and Chardonnay, harvested and grown in the same region and then made with exactly the same winemaking techniques. Again, there's a bit of variation, but our best wines here are as equal or better than the French varieties. And again, that's true for both Rimple and Wagga. So again, that gives us some confidence about the potential of these wines going forward. The other thing we wanted to look at is we've got 20 wines here. We want to really identify whether there are individual varieties in these 20 reds and 20 whites, which are generally more preferred across a range of tasters and also across different regions. And you can see what to get a handle on that, where you've got individual wine scores which are difficult to compare. What we've done is taken those wine scores and we've converted them into a ranking. We've used something called a standard competitive ranking system, where the top score gets a 1, and if there are then 2 scores which are the same, they'll both get 2, but then the third score gets a 4. So it tends to exacerbate differences in scores, where often you won't see a big difference in the scoring at an absolute level, but once you put the ranking in, it tends to expand the wines in terms of perceived wine quality. And I think you'll see here's an example of I've put in green those that have had a top 5 ranking in any one of the tastings. And then over here, I've also got the average ranking for all of these wine tastings across here. And you can see that some of the lines are consistently higher ranked. Sometimes they're highly ranked, but not in another tasting. But on average, you can see there's a number of lines. There's about 3 lines in the reds and also a line in the Tannat as a different style, which seem to be consistently ranked more highly across different wine tasters in different regions and between different wines produced from grapes grown in different regions as well. So we think going forward, these are the wines that we will push to be adopted by industry. But some of that adoption is already going on. So when we hold these wine tasting events, sometimes there are, as I said, there are wine company representatives, there are growers there. And after the wine tasting, they can see that a particular wine would suit a particular style that they want to produce or their company wants to produce. And so they've entered into evaluation agreements with CSIRO. And this just shows you that we currently have 7 arrangements with wine companies and growers who are evaluating a number of reds and whites. And the good thing about this is these are in a range of different regions, in the Riverland, in the Sunraysia, in the Murray Valley, and most recently in New South Wales. And so these growers are interested, or growers or companies are interested in evaluating. You'll hear a bit more about that. One of these companies is Treasury, and Ben's going to talk about the work that they're doing with a couple of the reds that they've selected. So I think we can give ourselves a big tick there that we have managed to produce what we're calling our first generation mildew-resistant vines, which have resistance from Muscadina rotundifolia. Now, as I said before, we haven't seen any evidence in our work of any sign of any powdery gainer resistance in any of our trial sites, even without sprays. But what we've decided to do is to mandate that when growers and companies are actually evaluating our grapes, that they actually use a minimum number of sulfur and copper sprays just to keep the level of inoculum down in the vineyard. And the reason for that is that we know, based on experience with other crops, that when you rely on resistance from single genes, that is then that may not necessarily be durable in the long term. To get durability, what you need to do is to combine genes from different sources and bring them together in the same organism, in the same plant. So that's what we're working towards at the moment, is to produce what we call our second generation no spray mildew resistant vines. And this is where we combine the resistance that we've already used in our first generation and combine it with resistance genes from another source to give more durable resistance in the vineyard. And we believe that those second generation vines will not need any spray at all. Now, when the French came to make this decision, they had also generated these first generation varieties with the bouquet breeding lines containing Run1 and RPV1. What they decided to do was to take advantage of the fact that the Germans had also been doing some breeding with a different American vita species. Oops, let me go back. And so these were the 2 varieties you may have heard. You've probably heard of Regent, and another one is Valaris, which is a white. And these contain a separate powdery resistance gene and a separate downy resistance gene. They crossed these to produce what they call their second generation resistant varieties. And some of those have actually been released commercially in France. Now, when we came to make this decision, we decided not to follow the French for a number of reasons. One is we didn't have this material in Australia at the time. Secondly, RPV3 is already broken down on a number of different occasions in European vineyards. So we wanted to steer clear of that particular downy mildew resistance gene because it didn't seem to be durable. And the third thing was we had no real information about whether Run1 and Ren3 actually have a different mechanism of resistance, or whether if an isolate was generated that could break Run1, it might possibly break Ren3 at the same time. And then you've got no advantage of having both genes there. So we took a slightly different route. And this was stimulated by a publication from some of our colleagues in the US at USDA, and they published a paper that showed that a wild Chinese species called Vitis romanesci actually also had very strong resistance, and that the mechanism seemed to be quite different to the Run1 gene. So we got together with our colleagues in the US and decided we really want to check out whether they do confer resistance to different isolates before we committed to actually introducing that into our breeding stocks. So without going into a lot of detail here, we carried out some experiments whereby we took plants containing the Ren4 resistance locus from the Chinese species, and we challenged that with an isolate that our American colleagues had discovered in North Carolina, the home of the muscadine grapes, which actually breaks the resistance conferred by Run 1. And when Ren 4 was challenged with an isolate that breaks Run 1, it maintained its resistance. So that told us that the Ren 4 resistance gene has a different mechanism and recognizes different powdery isolates than Run 1, and therefore it complements the resistance. So if by chance one of the resistance genes was to break in the field, it wouldn't break the second gene. So we think this is a much better strategy than using the strategy that the French used. However, that brings with it its own complications and challenges. The first was we didn't have any of this material in Australia, so we had to import some seeds of Vitis romanesci backcross 2 material containing Ren 4. We had to put that through quarantine. And this is only backcross 2 material, so it's only 87.5% vinifera with quite a large amount of the wild species still there. We had to carry out our own backcrossing to get to what we call the backcross 4, or 97% vinifera. Now you can see here, this is meant to remind me to tell you that a bit of serendipity here. Thank God we have a little bit of serendipity in science every now and again. And thank God we're allowed to do research which allows us to find that serendipity still. While we were doing the backcrossing, we actually discovered, to our amazement, that those plants that were inheriting Ren 4 and had powdery resistance also had downy resistance. And we characterized a new locus called RPV16. And this was a real boon because it meant that we could actually breed Ren 4 in and breed a second downy mildew resistance gene in at the same time. So the strategy for the second generation mildew resistance vines is as follows. We already have in the field our first gen selections that we've done wine tasting on, and we've chosen some of those good performing first gen selections that have Run 1 and RPV1. We take pollen from these in the field, and then we put them onto our female breeding lines that contain Ren 4 and RPV16. We then use marker-assisted selection again to pull out the second gen progeny that now have all 4 resistance genes, and then they can go into the field in the unsprayed block at Nuri to be evaluated. The problem with this strategy is that it means we can only use these parents to bring in varietal character. All of these are based on a Pinot background, but we can only, because we're relying on bringing in Run 1 and RPV1 from the first gen selections, we can only bring in these parents as the other character. However, the good thing about the second gen progeny is that they're segregating both for hermaphrodite plants, which will produce fruit, but some are also female breeding lines. And what we've been able to do in the last 12 months, some of those are now flowering, and these female breeding lines contain all 4 resistance genes, which means we can go and get pollen from any other variety, any other susceptible variety we want, and then use those as parents as well, which means we can introduce a lot more varietal differences and variation from only using the first gen selections. So we think by next year, at the end of the current project, we'll have over 500 second gen progeny in an unsprayed block at Nuri, and the first of those are flowering this season, and we'll start doing single vine evaluations on those and winemaking next year. And over the next few years, we hope to be able to carry that out on all 500 that we have in the field. So in summary, what are the opportunities we see with the new Australian mildew-resistant varieties? Well, the first one is obvious, and that's blending. High colour, low disease sprays required. That would be a great replacement for some of the varieties that are currently used for blending. There's also a possibility that some of these varieties may be of good enough quality that they could be a straight variety on their own, and maybe replace some of the existing varieties that are grown in the hot regions, which are going to struggle to produce colour with climate change. There's also an opportunity for new styles. So here's an example of the high colour wines, the red flesh varieties, crushed at much lower Beaumet levels. And you can see that they still produce, these are just with no skin contact, there's enough colour there to give a red wine. at low alcohol, which could potentially be low alcohol as well. There's also the possibility to use these grapes we talk about Australia's green credentials. Well, here's an opportunity to really push the green credentials, to not use any sprays at all. And I think that's a really good news story in the export market and to the local market as well. And the final thing is that these new varieties, unlike all of the varieties which are currently available, none of those can be protected. There was never any IP when they were first bred, and any IP that was available for more recent varieties is now expired. We can protect these new varieties, and they can be used only by Australian growers to make unique Australian wines that then could be sold in the export market. So with that, I have finished, and I'll hand it back to Robin to invite Ben to continue.
Thank you, Ian. Great presentation. I've got quite a few questions that I've written down. I encourage other people to start writing their questions in the Q&A section down below. Now I'd like to introduce Ben Harris. So Ben Harris was born and raised in the Adelaide Hills region of South Australia, where he commenced his involvement in the wine industry in 1993. Ben has since worked in several roles throughout Australia, New Zealand and France, including technical and management roles in McLaren Vale and Langhorne Creek, managing the Penfolds vineyards at Robe and managing the Wynns Coonawarra Estate vineyards. Ben's current role has him overseeing the technical strategy for the TWE viticulture team and managing viticulture in Victoria, Tasmania, WA, New Zealand, and the Wynns vineyards in the southeast of Australia. So I'm not sure how you have time to join us for this webinar, Ben, but if you are ready, I'd like to hand over to you and invite you to share your screen, please.
Thanks, Robin. I'll just share my screen. Has that worked, Robin?
Yeah, that's great. Thank you.
Excellent. Thanks, Robin. Thanks, Ian. Hi, everyone. Today, I'm just got a short presentation, and I'm really just going to be talking about why Treasury Wine Estates has been involved with CSIRO and these new varieties, and also where we're at, how we've developed and where we're at with assessing these new varieties. Okay. The reason, the main driver for us and our interest in these new varieties is sustainability. Sustainability is a priority for us. Our key targets around sustainability is to be, and our commitment is to be 100% renewable with electricity by 2024 and carbon net zero by 2030 for scope 1 and 2. We are focused on building a resilient business, and we're also focused on producing sustainable wine. So how do these new CSIRO disease-resistant varieties fit? Well, with sustainable wine, you know, a key drive for us is to reduce our energy and CO2 emissions. So the more spraying, or the less spraying we can do, you know, the less diesel we're burning and the less CO2 we're emitting. So for that, that's a major win. And it also reduces our chemical use. Building a resilient business. We are in a, you know, with climate change, we are seeing a warming climate, and we're also seeing a more variable climate. So how can we, I suppose, build some resilience into our vineyards that enables us to produce, improve, or if not maintain, wine quality and yield in a warming and more variable climate. And you know, the tannin and colour traits and the improved flowering that are bred into these new varieties play a role here. The other key driver for us is consumer preference. Consumers more and more are seeking solid sustainability credentials, as well as minimal chemical use. And so those that, sustainability is not an option anymore. Consumers are demanding it. And then on top of that, how can we show that we're reducing our reliance on chemicals in our industry? An example of this was a survey we did with our consumers in September 2020, which included people in the US, UK, Sweden, China, and Australia. This won't be a surprise to a lot of people. Sustainability, as I mentioned before, is a must-have, and to have solid sustainability credentials. But on top of that, and what we're seeing more and more people is minimal use of chemicals, pesticides, and fertilizers. So people will choose other wine products over another one based on sustainability, and they'll potentially pay more, or at least move towards the one that they are confident has less chemical use. So where are we at, and what are we doing with the CSIRO varieties here in Australia? So we're trialling 3 at our Makarranka vineyard, at the Treasury Wine Estates Makarranka vineyard in the Riverland. And our targets with the work we're doing in partnership with CSIRO are, how effective and our ability to reduce our chemical use through the reduction of powdery mildew and downy mildew sprays. This is driven by sustainability. It's not driven by cost. There is some upside in cost. In a hot climate like Makarranka, where we're not spraying a lot, so sort of 5 sprays and reducing those number of sprays, we're probably saving through reduction in chemical, reduction in diesel, labour, and requiring less tractors. We're probably saving around $155 a hectare. Obviously, if this was applied in a cool climate where you may be spraying 12 sprays a year, and you can reduce those sprays, the financial savings would probably go up to around $275 a hectare. So they're quite conservative numbers. There's potentially greater savings, but so our driver isn't financial. They're great to be saving money, but it is really about sustainability aspects. The other big one for us is chemical use, which I touched on, and our ability to reduce our CO2 emissions to help with our carbon neutral targets by 2030. So, the less tractor use, less chemical we're burning is, and so we'll assess our ability to reduce diesel as well. The flowering, the improved flowering, and also impact on yields is another thing they're assessing, and this is something that we're seeking. In a warm and hot climate, how can we produce at the quality levels we need the yields we're after? And that's what we're going to be assessing in the Riverland. But in a cooler climate, where flowering is more variable and we're more exposed to sort of cold, poor flowering conditions, and we see yield vary greatly based on flowering conditions, and also that variability in flowering that we get in poor flowering can lead through and have quality implications. So the ability to have more consistent and better flowerings is something that we're interested in. And we touched on it before, the climate change and the warming climate. So, the colour, the positive colour and tannin traits, and our ability to consistently produce or improve quality at desired yields is a focus. So, timeline and where we're at. So we started discussions with Ian and the CSIRO team in 2017. In 2018, we assessed 20 of the CSIRO varieties at the Barossa Sardi vineyard and from that we selected what we thought would be the 9 the best for us varieties, and we made small batch winemaking. We did commenced small batch winemaking for those 9 varieties. Our winemaking team assessed those 9 varieties in the small batch wine, and based purely on style and quality, wine quality, we select the top 3. And then we commenced propagation. So we worked with the Yalumba nursery to propagate out enough vines to plant a commercial vineyard at Markaranka in the Riverland. So that vineyard was planted in 2019. So we planted 2 hectares of each variety. So we'll get the first crop of those 3 blocks this year, and then we'll commence our commercial wine quality trials. We won't do it this year. We will have a crop, but as everyone would realise, in the first crop, it's probably not the best reflection of the potential of these varieties. So we'll start next year, and we'll, at a commercial scale, we'll assess the quality and the potential of the new varieties. And that's it from me. So thank you, and we're excited to see the potential.
Thank you, Ben. Great presentation. It's good to get a perspective from Treasury on why you went down this road, what you're considering, and excited to see the results after vintage 23. Thank you, and very interested to hear what you call these varieties as well. So I'd like to open it up for questions now. So we've got a few questions that have come through already. So Ian, could you please briefly explain the functions of the different resistance genes?
Okay, thanks, Robin. This gives me an opportunity to go back to my talk because I didn't put the acknowledgement slide up. But, can you see that?
Yes.
So
yes.
Please, Wine Australia, forgive me for not mentioning you in the initial talk. Of course, none of this could be possible without Wine Australia, and also wanted the people listed here, too many to list, but I want to particularly acknowledge Mark Thomas's contribution to this. Mark started breeding work, then decided to take early retirement, and so none of this would have been possible without Mark's initial input. Now, to answer the question, I was hoping someone would ask me this question because this is the bit I really love, the biology. So these are very interesting genes, and this is a demonstration of what they do. Both RUN1 and RPV1, what they do is the genes produce a protein, and the protein, if you like, is like it's like a sentinel. It's looking for an invading pathogen. When the pathogen invades, the pathogen secretes proteins to help it invade the cells. And these proteins, the RUN1 and RPV1 proteins, actually detect those proteins, and they initiate what's called programmed cell death. So here's an example of a powdery mildew spore which is germinated and formed an approsoria, which then pushes a feeding structure into the living cell. And this is 24 hours after infection on Vitis vinifera, which is susceptible, and Vitis vinifera that contains the RUN1 resistance gene. And you can't see much difference at this point of view. The green coloration of the leaves has been removed here. But 24 hours later, you can see that on the susceptible line, the powdery mildew has continued to ramify across the surface and continue to grow. But the line containing the RUN1 gene, the individual cells here have died. So essentially the mechanism is recognize the invading pathogen in that cell, induce that cell to undergo suicide, a suicide response, programmed cell death. And because these two pathogens are what we call biotrophic pathogens, in other words, they can only survive by taking up nutrition from the living plant. If that cell dies, they can't get any nutrition, so they can't grow any further. And this is exactly the reason we can't use the same mechanism against botrytis because botrytis is a necrotroph that actually lives on dead or dying cells. And so this type of mechanism would not work against a necrotroph, and that's why plants have not really come up with very good genes to control resistance to necrotrophic pathogens. So that's in a nutshell. They induce programmed cell death, and these cells sacrifice themselves for the sake of the rest of the plant.
And the other genes that you are working with now, the ones from the
Yep, the same, slightly different mechanism. I don't have a slide for REN4. It would take me too long to explain that, but both RUN1 and RPD1 have this same type of mechanism where they recognize a protein secreted by the invading pathogen and then initiate this cell death response in the cell that's actually been penetrated by the feeding structure of the pathogen.
Okay, just on the current recommendation to apply 2 sprays, so for powdery and downy during the season to protect those resistance genes. So when you have 2 resistance genes for powdery and 2 for downy, will you be still recommending that they apply some sprays for to protect those genes, or will you be? Will there be no?
We don't believe in Australia that. My understanding is that in France, what they've actually found is that when they completely eliminated sprays, they found pathogens that they weren't even aware they were controlling. So black rot and other things suddenly appeared when they stopped spraying with sulfur and copper. I think in Australia, we won't have that challenge. We don't have the same disease pressure. So I think we could realistically look at an opportunity to use the 2nd gen and apply no sprays at all in the vineyard. The thinking behind the 2 genes is that if one isolate was to undergo a mutation to allow it to overcome one gene, the chance that it would also mutate to overcome the 2nd gene at the same time is very, very low. So that's why you get much more durability by combining genes of different mechanisms together. Okay,
great. Thank you. We've got a question about genetically modified varieties. So Ian, could you see genetically modified resistant versions of traditional varieties being used in Australia in the future, or only the new bred varieties described today? Are any other countries growing GM varieties?
No. To the best of my knowledge, there's no GM grapevines used in any wine production in the world, and I don't see that changing in the near future. Obviously, we see these new varieties as a potential replacement, but I am aware there's a large part of the industry still very much in love with their classic French varieties and would love to have resistance in those varieties rather than planting new varieties. The only opportunity we see for that is a completely new technology. Most of you would have heard of gene editing. So gene editing used in combination with a new propagation system based on protoplasts, what we call DNA-free gene editing. So this is all very new. It's only been really demonstrated in a couple of labs in Italy in the last 12 months. It may be possible to produce genetically altered versions of existing varieties, if you like, clones using gene editing. That's something for the future. But the standard, the GM varieties that I have sitting up in our glasshouse that will remain there forever, which are the classic genetically modified organisms that contain foreign DNA or marker genes, I don't see that they'll ever be used for wine production.
Okay, thank you. How will the new varieties be named, and when is this likely? Perhaps Ben, or Ian, you're about to speak.
No, I was going to say, actually, of all the things, this is probably one of the hardest things to come up with new names. So we're doing some internal surveys and thinking of different opportunities. TWE have got 3 varieties, and I know they're considering what their naming might be. Ben, I don't know whether you want to talk about how TWE might go forward on this.
Well, we're we're working through it. It's a very good question. We're looking at a range of ways to name it. We haven't come up with names yet, so we have an opportunity to come up with the 3 varieties, 3 potential names that we then run past CSIRO and Wine Australia for approval before they're potentially registered. It's actually really difficult to name varieties. It's actually much harder than we were anticipating, but we've got a team working on potential names at the moment. But yeah, we're not there yet.
If anyone has any good ideas, feel free to send them to me. We'll take any suggestions we'll consider.
Oh, I gave you a very good suggestion.
Yeah, I wasn't convinced by that one, Robin.
So we've got a great question about testing these varieties in cooler regions that have more, as you noted in your presentation, Ben, they apply more sprays to protect themselves against mildews. So the fact that there's more potential to save money, but also more potential to reduce chemical use. So you have looked at orange, but are you looking at any other regions? And my other addition to that question is, do you have the wine quality data from orange? I don't remember seeing it in your presentation.
No, and you're right, I didn't show it. We've only just made the first vintage this year, so I guess orange was a bit of an afterthought. We were focusing more on Arimple and Wagga, and I guess that was also partly because the bulk of the Australian industry is in the hot, warm areas. So in terms of adoption and impact, we were probably more focused on that. But we have now established, thanks to donation of land from New South Wales DPI, we have established a vineyard in orange. We had the first vintage this year, and it's we had a bit of trouble getting it to ripen. I'm sure a lot of growers in orange had that same problem this year. But again, at the moment, we've not seen any major disease issues in the unsprayed vineyard that we have in orange. And as I said, we got the first vintage this year, and that was only just tasted a couple of weeks ago. So we haven't really analysed the results yet. And what we're looking forward to doing is taking the wine that we made in orange and having some wine tastings with growers and winemakers in the orange area to get their impression of how they see these varieties would go in that region.
Great. And just an addition to that question, my thoughts were, have we missed an opportunity by selecting the 20 varieties that we're planting in Nuri and looking for qualities that increase the flowering of these varieties? So in cooler climates, is it more important that we increase the number of bunches, but we want more open bunches so that they're not more susceptible to things like botrytis? So do we really need to be selecting varieties that are suited to warmer climates and then going back to those 1200 and selecting varieties that are suited to cooler climates?
I need you on the Wine Australia research board, Robin. It's really a question of money. That's say that's all it is, but the reality is, given Wine Australia now have endless amounts of funds to put towards this, so you try and get your best bang for your buck. And so you are right. Ideally, when we do those crosses and we put those 1200 vines out, we put them all out in Nuri and evaluated all of those, and that's convenient because we can drive to Nuri and we can actually do the small-scale winemaking ourselves. We did anticipate, we did think about with the second generation, planting them both in Nuri and orange. And so we could evaluate the single vines exactly as you had predicted, because of course, once you select for conditions in Nuri, you may have thrown away the things which are going to be really good for full climate. Yep. But the realities of having to sample in orange and do small-scale winemaking, it just became impossible unless we had a huge budget to establish another facility in orange. So I think if we were given more money, that's ideally what you would do, but it's funding which is limiting, really.
Or is there opportunity to work collaboratively with other organisations in these cooler climate regions to do the work for you on the ground?
That's a really good idea, and I suppose that's always an opportunity. This thing, it's not cheap to do this, I'd have to say. So we're talking significant amounts of money, particularly when winemaking becomes involved as well. But in an ideal world, yes, you would not apply any particular selection. If you wanted to find something that was going to be adapted to a cool climate, better adapted, you would actually do the evaluation there. But I'd have to say, from the initial results we've got, those varieties which are doing well in the hot climates also seem to be doing well in the cooler climates as well.
And you said you use DNA markers to select the crosses that have the better flowering. So can you go back to that step and select the varieties with more open bunches?
Right. So I probably didn't explain that very well. The marker we use for flowering allows us, because when you cross grapevines, they will actually segregate and produce either female flowers or hermaphrodite flowers. Now, female flowers obviously can be pollinated in the field. There's lots of pollen, but they won't necessarily be as efficient as a hermaphrodite flower. So what we were doing there was particularly only selecting those progeny that had hermaphrodite flowers, but we don't have any markers at the moment that could predict how many flowers you're likely to get. That could be something in the future and will be a really useful trait to have.
All right, great, thank you. We've got a really great question here about the potential to search for genes that have allow vines to have greater tolerance to extreme environmental factors such as heat or drought.
Yes, so I guess thinking about climate change, it's something we've been thinking about as well, is that we think we've got a pretty good handle now on disease resistance for the major pathogens. Obviously, detritus is still an issue, and there are other pathogens, trunk disease and the like. I think the big challenge going forward that we all know about is climate change and how these vines are going to, how are we going to find varieties that are going to stand up to the heat waves and high temperature conditions. And I think that is a completely separate selection process. And I don't think we've quite worked out, those traits are likely to involve multiple genes, not single genes. So one of the advantages of doing things like disease resistance or salt tolerance, some of these traits are inherited mainly by one major gene or locus. So it makes the breeding feasible. Once you start looking at things like drought tolerance or heat tolerance, that's likely to involve multiple genes, each having a small additive effect. And that becomes actually really challenging when doing breeding, because the numbers of seedlings you have to produce to get that one that's got all of those genes is mind-boggling. So I'm not sure how, and the other challenge is, how do you actually then screen the progeny to know whether it's actually going to stand up as a mature plant in the field? So we have to do a lot more work to try and work out a proxy for perhaps evaluating seedlings in a glass house or the plant accelerator we've got on the Wake campus here, and see whether that could in any way relate to heat or disease, heat tolerance or drought tolerance in the field as a mature vine. But there's a lot more work to be done there, but it's really going to be the major problem going forward, I think.
Thank you. And Ben, a question for you. Are the Treasury varieties red or white or both?
3 red varieties. So we're only trialing reds.
Thank you. And Ian, what is the realistic time frame for the release of the double resistance varieties?
Okay, so looking at the way the process goes, essentially to do single vine evaluation, it's going to take at least 4 years. The plants have to grow, put some fruit on to evaluate them. Unfortunately, there's no getting around the fact that once we take that single vine, we now have to put it into replicated trials of probably 12 to 24 vines to get enough fruit to evaluate it on a medium scale. Unfortunately, that means there's no way to make the plant grow any faster than it currently does. So that means another 3 to 5 years to get at least 2 or 3 vintages to then evaluate. All up, it's about 10 years from the day you put a plant in the ground from a crossing to have something that's then you could confidently say, based on viticultural characteristics in a number of regions, based on wine quality, this could now make a good commercial variety. I wish it was faster, but there's nothing we can do about it.
Okay, thank you. There's a question, are any participants in WA doing similar trials of the new resistant varieties?
No, not at the moment. We welcome any opportunity. Of course, the problem with sending stuff to WA is it then has to go through quarantine again. And so there's a bit more of a delay. Well, that's my understanding. So we have had inquiries from people interested. And again, if there's people who would like to trial these in WA, please contact me and we'll try and work out some way to meet that request.
Okay, thank you. Then this is a question from me, are you able to speak to the challenge that the red-fleshed varieties present in when you're doing, let's say, virus assessment or
Yeah, no, that's a good point. Like part of the red flesh varieties, which is sort of related to the improved colour, which is, you know, very beneficial. We did have a minor panic when they were young vines, when they all started turning red and look very similar to leaf roll symptoms. So I suppose the challenge is there as part of our, when we put in new vineyards, we monitor them closely initially for virus, and if there's any vines that are potentially have leaf roll, we remove them immediately. So, which we can't really do that visually with these new varieties because they do display red leaves and symptoms that look like leaf roll. But it's, to be honest, it's no different to white varieties that don't express leaf roll symptoms, so they can have leaf roll. It just means we have to do more testing. So we have to do strategic testing just to be sure that there's no leaf roll. But we have not found any leaf roll in any of these varieties. It's just part of our standard practice.
Okay, great. And have you noticed any changes in or any difference in the growth habit or the vigor of these varieties compared to other varieties? Have you had any challenges?
Not really. The biggest challenge was how to propagate from, you know, a limited number of mother vines up to enough vines to plant a commercial planting. So that was probably the biggest challenge, which is, you know, 2 hectares gives us a decent parcel size to make sort of a decent volume of fruit to assess them. So there are slight differences at the moment between the 3, but it's sort of, they're on 3 different sites. So what sort of environmental and based on the site and what does actually do the variety, we're not sure yet. And we don't have I suppose, a traditional variety next door to compare. So, and it's very early. So we only planted them in 2019. So I think we need more time to really assess if there is differences in how they perform in the vineyard and, you know, water use, canopy size, quality. I think that's still to come.
Great, thank you. I have a million questions for you, Ian, about genetic editing or gene editing, but I think that's for another time.
Okay. Yeah.
I'd like to thank you both, Ian and Ben, for your contributions today. It was yeah, a really interesting webinar. Watch this space. We'll have to get you back for an update in a couple of years' time. So just a reminder that this webinar has been recorded and the recording will be emailed out to you. Our next webinar is next Thursday. Next Thursday, the 28th of October, and it's on carbon sequestration. So I'm really looking forward to hearing what Sharon Harvey, Dr. Sharon Harvey, Professor Tim Cavanaro, Professor Robert White and Ollie Madgett have to say on the topic. So if you'd like to register, just jump on the AWRI website and look for the webinars in the what's it called, Jess? In the area where I cannot remember. Anyway, just Google webinar AWRI and then you'll find it. Thank you again, Ian and Ben, and look forward to seeing everyone again next week. Thank you.
Thanks, everyone.